So what I'm going to talk about today is how our memory influences the decisions we make, and also how our prior decisions influence the memories we encode. And the best analogy that I like to show when talking about memory-guided decision-making is this classic New Yorker cover from the 1970s that shows a New Yorker's perspective on the world, which is centered on Manhattan and very little else. So if you asked someone from Manhattan their favorite spot in the world, they'd have a hard time maybe choosing between Greenwich Village and Central Park, whereas someone from somewhere else in one of these more enlightened parts, sparse parts, perhaps even to the north, might have a hard time choosing between their favorite beach in Barbados as their favorite place, or going to Paris, for instance. And so what I'll talk about today is, a lot of you probably already know a lot of this, is give you a little background about cognitive maps and how we construct our mental maps of the world. And then I'll show that how we rapidly make multi-step decisions in novel situations. It's determine whether cognitive maps help us afford this type of flexible learning and decision making. And then I'll go through the neural dynamics underlying goal-directed imagination and mental exploration. And lastly, I'll conclude by presenting a new study showing how cognitive map-like coding can help us understand also how we learn about new people and social learning, and not just spatial navigation. And then hopefully I'll have a bit of time to discuss how Lethbridge can facilitate my future research. So first, just a little background on cognitive maps. So the idea of cognitive maps really started in the 1930s and 1940s when Edward Tolman found that rats exploring a maze to locate food found that it wasn't a function. They learned it not as simply as a function of previous experience, but they encoded transitive relations between locations that they never experienced. So when they opened when a detour was created or a shortcut was opened in the maze, the rat could immediately figure out that shortcut to find the shortest route. And although they had some difficulty replicating these behavioral findings, the idea of cognitive map theory was very important in the Nobel Prize-winning discoveries of play cells in the hippocampus corresponding to particular locations in an environment that a rat explores, grid cells, which correspond to the whole environment that a rat explores in the entorhinal cortex, medial entorhinal cortex, and also head direction cells that correspond to the direction, the environmental direction that the rat is facing in subiculum and retrosplenial cortex. And more recently, particularly with grid cells, these similar responses have been observed in humans, whether it's grid-like coding in the medial entorhinal cortex and also surrounding regions, which I'll also discuss later, or directly recording single units in pre-surgical epilepsy patients that have monitoring electrodes in these regions. And instead of a rat exploring an open box, these people would just be playing like a Navigate spatial memory video game. And these are the main brain areas that I'll be focusing on today, just as a reference to refer back to. Although here the entorhinal cortex extends a bit further back than it probably actually does. But that's not particularly important here. So let's explore the term mental exploration. And this is something that's really interesting in terms of how hippocampal coding and the coding of single units in the hippocampus might pertain to deliberative and evaluative behaviors. And I like referring to it, even though I'm in Toronto, referring to it as mental exploration instead of mental time travel, because it's searching for a novel sequence of actions in order to achieve a goal, or as Hopfield would put it, the famous computational neuroscientist, or searching for an appropriate neural activity sequence that may never before have been experienced. And this is directly related to hippocampal place and grid cells, because the phenomena of theta phase precession, coupling between the exploration-induced hippocampal theta rhythm, couples to the firing of place and grid cells. And this is one of the few examples with neural oscillations of a behaviorally relevant neural firing relating to an ongoing oscillation. And as a consequence of this theta phase precession, forward sweeps of place cell trajectories occur when a rat approaches a choice point or a bifurcation within a maze. And so numerous people have thought that this might be a single unit correlate of planning or mental exploration, even though physiologically it's just the firing fields, the overlap of firing fields allow future locations in either direction when choosing an option and choice points. But you can think of this as maybe a putative single unit correlate of planning, even though it's hard to actually probe performance-wise on a rat in a T-maze. On the other hand, in humans, we have although we're not, for the most part, we're not recording on a single neuron level, we can actually interrogate this internally guided behavior. And so this centers not just on the medial temporal lobe, but a whole set of brain regions called the default mode network, including the medial prefrontal cortex, medial parietal areas like the retrosplenial cortex, and the hippocampal formation that show correlated activity at rest, but also relate to imagination, planning, spatial navigation, and episodic memory. And looking at how those regions have related to those behaviors, in a recent review, we formulated the hypothesis that neural computations related to spatial coding could also facilitate flexible learning and decision making based on an internal model of the world. And perhaps these default network regions are representing an internal model or template upon which we make novel decisions or learn novel stimuli very rapidly. So speaking of learning things very rapidly, I'm going to explore how we rapidly make multi-step decisions in novel environments in terms of spatial planning. But first I'm going to look at reactivation of learned representations and see how the endogenous influence of hippocampal place cells and the endogenous replay of neural activity could correspond to updating this internal model of the world and updating this internal map. And so I'll talk about another phenomenon that many people have studied here for many years. Hippocampal sharp wave ripples are the offline reactivation of spatial trajectories. So I highlighted the human hippocampus and a sharp wave ripple, which has occurred during restful and deliberative periods and have mainly been measured in rodents. And there are large deflections, you can see the deflection here, or sharp waves in the hippocampal local field potential that co-occur with high frequency oscillations lasting at least 200 milliseconds, called ripple oscillations. And these ripples are associated with memory consolidation, since reactivation of place cell ensembles occurs during ripples, and this is seminal work by Bruce McNaughton here, and others. And that the behavioral correlates have been a bit more unclear and a bit less definitive of what the correlates are, but the number of ripples after learning have been observed to predict later memory performance in both rodents and humans. And electrical stimulation of the hippocampus after learning interferes with consolidation. And related to this, in a recent, well, not so recent now, about 6 years ago, Nikos Logothetis published a study where he invented a technique called neural event triggered functional MRI, where he simultaneously records the hippocampal local field potential in anesthetized macaques while they're also undergoing functional MRI scanning. And so this allowed him to look at spontaneous events in the hippocampus, whether they be ripple oscillations, so not the actual activity of single units, but just the oscillations, ripple oscillations or sigma events or low frequency gamma events, and look at how brain dynamics in terms of the bold signal occur across the whole brain at the same time. And what he observed, the main result of his study was that fMRI activations, other than the technological wonder of getting that to work, was that fMRI activations in voxels across the neocortex and deactivations in subcortical regions occurred specifically after ripples, but not other hippocampal events at other frequencies. And that voxels in core, and what I noticed when reading this paper, is that voxels in core default network regions showed dramatic increases. But this wasn't clear. What wasn't clear, even though these regions usually show correlated activity at rest, was whether these events were inducing some sort of phase reset or uniformly influencing this resting state network, or this was just diffuse activation across the whole neocortex. And luckily for me, I was in Barcelona working with the computational neuroscientist Gustavo Deco, and I had access to the data to test this particular hypothesis. And in order to test this hypothesis, I had these specific questions and aims. First was that are hippocampal sharp wave ripples influencing ongoing resting state fMRI activity in unitary resting state networks, or just diffuse transient activation across different neocortical regions? And secondly, are they influencing some resting state networks, like the default network that I mentioned earlier, more than others, versus maybe like a visual network or correlated activity in between lateral prefrontal areas and posterior parietal areas, for instance. And I was really excited about this, especially after reading Artur Luzak and Bruce McNaughton's review with Kenneth Harris in Nature Reviewed Neuroscience, as an opportunity to connect two disparate literatures of potentially relating behaviorally relevant circuit level activity to resting state fMRI fluctuations at the macroscopic or network level, which has been relatively ignored between the two communities. First, because at the circuit level, you have sort of a ground truth measurement that you can relate to behavior, whereas the fMRI community is more focused on dynamics and not seeing how it really relates to cognition or behavior, especially at the circuit level. So what we did was we used an independent component analysis on these different macaques. We had two anesthetized macaques who each had 25 10-minute resting state MRI sessions while they were also recording from the hippocampal LFP. And so we could use it, ICA, to separate these regions and to have a default network, a cerebellar network of areas that showed correlated activity with cerebellar or whatever strong component there was, and other regions across those 25 runs in each of the animals. And here's just what I'm showing here, which doesn't show up as well on the screen for whatever reason, is the ongoing bold signal across a typical 10-minute session. So you see this 0.1 hertz fluctuation of this whole network, and this is the correlated activity of this whole network. And these red dots here are the onsets of what you term ripple events. So in the macaque, I think it was around about 80 to 120 hertz. And a key thing to keep in mind that they didn't emphasize in the Logothetis nature study was that the ripples, a primary reason why the ripples might be influencing this network level activity was that the ripples, at least that they measured, and I'd be interested to hear other people's experience with rodents, was an order of magnitude higher, the amplitude, than these other electrophysiological events. So if there was to be propagation of activity, that would probably be the primary driver. And so this allowed us, having these resting state networks just together in one signal, average across one signal for each session, there's multiple brain regions, across one session, allowed us to just do a linear regression analysis on different networks with these different events. So we'd have the ripple events, these sigma events, and these low gamma events, and then we could look at the next most robust network, which luckily didn't correspond to, isn't normally associated with episodic memory consolidation, ventral somatomotor areas that were both robustly observed across both sessions, and compare whether this was network specific and whether this was event specific. And what we found was the only network that was consistently activated after ripples or showed increased activity after ripples was a default mode network. And this was particular to ripples. And here's the time course. So you see some variation between the monkeys, but the only consistent activity was there. And then we looked at some other less robustly measured networks across the multiple sessions. So a frontal parietal network, occipital temporal, primary visual, and ventral somatomotor. And saw that the hippocampal sharp wave ripples selectively activate the default network. This is not to say that they're the only regions being influenced by the default network, but that they're the primary target of the propagation of neural activity sent from the hippocampus. And currently, we don't have specific results about this yet. I'm still working with Gustavo Deco to look at neural mass models of this ripple propagation. So if we put a high amplitude transient activity in the hippocampus versus another region, other control regions, given that using anatomical information that we know about the macaque and the human brain and the anatomical connections between the brain, how would that activity propagate to other regions, especially if you simulate ongoing bold activity? And that can allow us to better understand the dynamics and how those dynamics might be perturbed. So just to summarize this first study, we find that hippocampal ripples, but not other events, correlate with increases in ongoing default network bold signal. And the default network is the only resting state network displaying consistent signal increases after hippocampal ripples. And this is very exciting but very preliminary evidence of cognitively relevant spontaneous neural firing influencing ongoing resting state fluctuations in the fMRI signal. And the potential mechanism that we're working to have a phenomenological model of is high ripple amplitude, the anatomical distance between the hippocampus and other default network regions like the medial prefrontal cortex and medial parietal cortex, between hippocampus and other core default network regions, allow for primary information exchange between these regions. And this is very preliminary evidence that these default network regions, so medial prefrontal cortex, medial parietal lobe, and hippocampal formation at rest, could reflect maintenance of map-like representations. However, better links with reactivation of learned map-like representations at the single neuron level and neural ensemble level, and novel hemodynamic response functions, along with high-resolution tract imaging in all these species are still needed. So I'm going to now focus on spatial planning. So how do we make a rapid sequence of spatial choices in a novel environment without seeing that environment before? So this is not to say, and this has been very poorly understood in the decision-making literature, because most research on the interaction between memory and decision-making is mainly focused on the storage of value representations. So how we remember something that's very rewarding versus not very rewarding. And in parallel, a model of sequential decision-making or even mental simulation are focused on investigating all possible options after either rapid or extensive deliberation. So for people that study robotics and artificial intelligence, this could be seen as a tree search of searching every branch. But what I was interested in is how do we restrict that search? So we might not make optimal decisions, but how do we have a first hunch about a decision that we're going to make and restrict that search space? So just as an example, if we're choosing between two places to go to ice cream after lunch, how do we deliberate between those two places without having to exhaustively look at all the menus and focus just on the ice cream, decide between A and B first, and then not have to deliberate about all that B has to offer and just focus on the more difficult perspective decision of choosing between vanilla and strawberry ice cream? And there are some clues on looking at spatial planning in novel environments. So I'm happy to answer questions about this study, the fMRI study, after the talk, but just for the sake of time, I'll just summarize it quickly. Is that in an fMRI study, we observed that the anterior prefrontal cortex and the hippocampus responded to a subset of potential choices, i.e. a restricted search, while lateral prefrontal cortical areas and the anterior cingulate and posterior parietal cortex responded to all choices when you had to make multiple choices within a maze, when you're visually interrogating a maze and a novel maze each time. And I'll explain the task on the next slide so you get a better idea of what I'm talking about. And so the idea here And the idea for looking at the electrophysiology data that I'll present in a few slides is, does this reflect a choice point location update? So what I'm interested in here is what we know about theta phase precession of reflecting a location, the theta rhythm reflecting a location update when a rat is exploring. Can we look at this in a more abstract dimension? So if we're doing a multi-step decision, and my favorite example is actually from social psychology. There's this classic social psychology study from the 80s where it's a really simple study. It's you ask someone what they're doing, and if on a multi-step task. So let's say they're preparing dinner. If they're cutting cucumbers and that's going fine, and you ask them what they're doing, they'll say, "Oh, I'm preparing a stew." But if they're having difficulty with that particular sub-action, they'll always report the sub-step that they're stuck on. And I wanted to relate theta phase precession and spatial planning to that. Can it reflect a location update on these, of a step update within these multi-step plans? And so how we did this is we leveraged our implicit knowledge of exploring the physical world to avoid extensive training and allow us to do sequential planning problems in a spatial maze that looked like this, and I'll show the actual trial structure later. And what participants did here is plan the shortest route from the red to the green, and they had to maintain the directional choices they made on the way. So the key aspect of this task was they knew always the initial choice they had to make of going left or right here. It could be left or forward, depending on the configuration of the maze. But finding the further choice point, the second choice point, this more second choice point along the optimal route. And once they found that, then they could kind of break down the decision into the second perspective choice. So here it would be left and then right, but there was also these other paths. It might not be clear on the screen here, but there's 4 paths to the green goal. And then they'd be probed on one of those choices. And using spatial planning, and why I'm interested in spatial cognition is not particularly because I'm interested in spatial navigation, but using spatial planning allows us to parametrically manipulate by changing path length. So you could have an easy initial decision where the path length difference between choosing left or right is very small or very hard, or it could be a large path length difference and very easy. And at the same time, at the second level of the tree here, this 2-level tree search, you could have a difficult decision or an easy decision. So we could counterbalance the influence of those 2 effects to see what was particular to the difficult perspective decision. And we could do this more in terms of, instead of a virtual navigation task of people playing video games and taking up a lot of time for fMRI design's sake, more in the style of a perceptual decision-making task, where they're just visually interrogating an overhead view of a maze. And relate this to the fMRI study that I recently published that found hippocampal and anterior prefrontal activity perspective signals related to this task. So here's the task. So first, subjects look at a maze like this and have to find the shortest route from the red to green and the 2 directional decisions they have to make along the way. So it's left or forward here, and then since this is the ideal route, it's a bit hard, so you might not have been able to tell. They're never tested on the wrong second decision. And then left or forward here. And then after 3 seconds, it highlights either the initial choice or the secondary choice, and briefly, and then it zooms in on whichever one it highlights, and they have 1,500 milliseconds to make a choice. So they really need to have a choice in mind, because it will go too quickly for them to make an accurate choice or to guess otherwise. And it just goes through, and then it goes to a new maze after that, and so forth. And we do over 200 novel mazes at varying levels of difficulty there. So since this is a very perceptual-like spatial decision-making task, sequential decision-making task, I wanted to also present this in terms of the potential saccadic eye movements that I hope would correspond to the search pattern in this task of a restricted search space versus exhaustively exploring every single option to find the shortest choice of the goal. So I was interested in terms of saccadic eye movements was first locating both choice points, perspective choice points, but then restricting it to only deliberating on the most difficult optimal choice point. So they may not get the secondary decision correct, but they'll at least restrict the search area, knowing, oh, I need to go left, I'll focus on this quadrant of the maze. That's the type of cognitive map-like computation that I'm interested in. And so to test this in terms of the hippocampal theta rhythm, we tested 22 healthy volunteers measured by non-invasive whole-head magnetoencephalography, which is like electroencephalography, except it just measures the electric fields measured by magnetic currents in the brain. And a pre-surgical epilepsy patient with hippocampal depth electrodes from a clinical collaboration I had in Barcelona. So that way, given the difficulties of source reconstruction and estimating the origin of scalp level activity, we would have a prior on this particular frequency and ongoing activity and a power spectrum from the hippocampus during this task of a few patients that would perform this task, and use that to restrict our analysis in the 22 healthy volunteers, where we had a more statistical power and a wider variety of options. And just like in the fMRI study, we found a significant correlation between the reaction time, so the amount of time it took to make that decision once they were zoomed in on that choice point, and performance, and whether they got that correct. And so here's just the electrode locations in the epilepsy patient who was a mountain climber and was young and was better than most of our healthy volunteers at the task, performing close to ceiling. And it showed this nice, strong 2.5 to 3 hertz, so it's lower frequency than in the rodent, but I can discuss that afterwards about the differences between the human hippocampal theta rhythm and the rodent hippocampal theta rhythm, if anyone's interested. And with 2 recording sites, one anterior and posterior hippocampus. And they have very quick reaction time. So after that planning phase, it only took them on average 360 milliseconds to make a decision. And locating this 2.5 to 6 hertz theta rhythm from the human hippocampus in the epileptic patient, we found that the reaction time activity during this planning phase, so I forgot to mention that we focus our analysis just on this 3-second visual search when they have the overhead view of the maze before they make a choice, and see what corresponds to that. And so correlating the activity during this 3-second search with reaction time, we found a negative correlation between, that's why this is blue, between hippocampal theta power and reaction time. So the higher the hippocampal theta power during the initial planning period, the quicker they made a subsequent decision, which also correlated with performance. And we see this very clearly throughout the whole planning phase, from 2.5 to 6 hertz in the epilepsy patient. But additionally, across the 22 healthy volunteers, we see this in the same region at the scalp level, and also when we do source reconstruction for reaction time. So one explanation of this, and this is very common with neural oscillation because a lot of the origin might be epiphenomenal, might be, oh, this just might be them performing a task correctly and just not task specific. So we looked at it and compared these sequential planning when they had to make 2 choices or plan 2 choices versus when there were other control mazes that we had that I didn't show you earlier, where there was only 2 routes. So there was only a decision to make it the red, and that was it. They already knew what they would make a choice on. So if it was just performance general, then you might just see that related across both trials. But what we find is we only find a strong significant negative correlation for the sequential plan. So a restriction of search space only for the sequential planning, and not just a general easiness measure. And just to show, which isn't as clear in the intracranial patient, but at the source level in humans, we see increased hippocampal theta power just for baseline for sequential planning versus baseline, to show that there is some ongoing hippocampal theta rhythm in healthy human volunteers too. And in a study that should come out in the next couple of weeks, we're working with Carl Friston, we tried to develop a simulation of how a myopic agent could do a similar task that could only look 2 moves ahead, and look at how we prospectively evaluate choices during sequential decision making in novel environments. And what we found was that this, and you can run, well, just email me if you need. I don't know why the font came out so poorly on here, but just there's a demo you can run on SPM if anyone's interested in playing around with it, and it's all in MATLAB, so you can edit the script to whatever you want. And what we find is that a simulated myopic agent's saccadic eye movements first gravitate towards open areas or subgoals before moving to a goal location. So when the simulated agent has no idea and can only look 2 moves ahead in a direction, it gravitates towards the most open areas in the maze. That's what we took away from the study. And what we're doing now, I'm supervising a clinical research student, is to look at eye movements and having a normative account of how saccadic eye movements might correspond to this decision-making process, which is one advantage of having this perceptual decision-making like task instead of a virtual navigation task where they can be looking and moving all over. So just an interim summary. We find that hippocampal theta power might relate to a location update within a decision space. Here it's spatial, but you can envision maybe how we could derive tasks that don't have as strong a spatial component. And that saccadic eye movements of a simulated agent in a similar situation in a novel environment gravitate towards sub-goals and can effectively solve the maze. So now I'm going to present my last study of using cognitive maps to understand how we learn about other people. So this is looking at like the phenomena of path integration, how we combine first-person information and environmental information when we update our position within an environment and know where we are in an environment, and see how we can use that same sort of computation to learn about other people. So learn about someone's preferences relative to someone we know and relate it back to ourselves, or vice versa. And the key concept when we're doing these sorts of coordinate transforms is ordinate versus metric coding. And so what we find is ordinal coding is fairly well studied in neuroscience. I like strawberry ice cream more than chocolate ice cream, and then my third favorite is vanilla ice cream. But what's much less studied and just as important is this metric coding. How much more do we like strawberry than chocolate ice cream, or chocolate versus vanilla ice cream? We could like strawberry and chocolate relatively the same, so there's not as much difference between first and second. Or strawberry could be the ultimate favorite, especially if someone's going shopping for their spouse. This can be a very important computation to make, of knowing these decision thresholds. So I was interested in that, and probably you can look at that across most of the brain, but why I wanted to really look at hippocampal function and see whether there was allocentric reference frame during decision making was how we related to other people. So if I know my friend is faster than Larry David and slower than Usain Bolt, neuroscience can tell us the neural correlates of that. But that doesn't, in real life, that doesn't tell us very much, because the person could be an Olympic sprinter that lost to Usain Bolt in the finals of the Olympics, or just barely faster than Larry David. So it's these type of information and these type of transformations that I wanted to look at that have not been that well explored in the cognitive neuroscience literature. But what we do know is that the amygdala and the anterior or ventral hippocampus encode ordinal structure in a domain general manner, so hierarchies. If I know this is the most important thing and then this is the most important thing, that's been observed, and especially in social situations, you can really evoke those responses. But where might we metrically encode decision variables, and why is it important for social learning? I have the hippocampus right there, so you can probably take a guess where I think it might occur. And why I think it's important is that order judgments will rely on transitive inference, which can be used with ordinal structure, but also invariant reference frames in order to relate other people's preferences with our own. And I think, and some clues of why the hippocampal formation might be involved is the hippocampus is linked with coding current spatial distance from goals, which was always a finding that interested me, because you see this in rodents and humans, more recently with rodents actually. But in terms of the hippocampal role in memory, it doesn't make a lot of sense of like, why would the posterior hippocampus code how far away you are from a desired location, while maybe the anterior hippocampus your proximity location. If you're trying to relate that to decision making or something more general, it doesn't really make too much sense. And to this end, parasubiculum and entorhinal cortex and retrosplenial cortex are also important to allocentric or invariant coding of the environment during spatial cognition. And in particular, what particularly interested me the most is that subicular and entorhinal regions signal direction of an intended goal in an invariant manner, and can represent, especially the entorhinal cortex and its role in grid cells, can represent spatial information at multiple levels of complexity and multiple levels of scale, versus the hippocampus, where you're seeing these distance signals at continuous and more discretized dimensions. So I was interested in probing that relationship and see how we can look at that in more abstract cognitive realms. So the two main questions in this study that I'll present to you is there a hippocampal-based metric representation of personal preferences or rankings during decision making? So shifting first and second and third preferred things versus that configuration of first, second, and third? And if there is, how do we rescale metric representations to our own preferences versus someone else's? So essentially, just absolute versus relative coding. So how I tested this and how I tested people and probed this with people's metric representation of social knowledge was I had 24 participants who were mostly UCL students, choose a close friend of the same gender but with a very different personality, and ideally living outside of London. So that way they would have, I could test them on a variety of scenarios where they'd have likely different ratings. And so outside of the scanner, before doing the actual experiment, they gave likelihood of ratings from 1 to 9 on a 0 to 10 scale for themselves, this aforementioned different friend, and also a typical individual, or what their concept of a typical individual would be. And we call that Mr. or Mrs. Normal, depending on their gender. On a scale from 1 to 9 for 100 different everyday scenarios. And that we try to make them as banal as possible, so how much someone likes spicy food, or how likely they are to send a message using, yeah, you can't really read that, how likely they are to send a message using mostly emojis, things like that. And then after they gave those ratings for each individual, and sort of hopefully constructed a mental number line of these personal preferences or of personal knowledge, they also gave confidence ratings on a scale of 1 to 5. So if you had no idea if your friend likes Sudoku or not, you could put you have no idea, and even though you rated a 6, we could use that confidence rating as a standard deviation measure to show how noisy or how stable that representation is versus something that you knew for sure that your friend hates romantic movies or something. And also in parallel to make sure these confidence ratings, to give some ground truth to these confidence ratings, after scanning they had to give likelihood ratings for these other individuals, again, to get a consistency score. So similar to what you'd have for a distance error for spatial memory, where on the number line their ratings were. And so, since there's over 100 scenarios, we didn't want participants to remember 3, 5, 7 for every trial, but just the rough location of each individual on each on the number line. And then here's the task, and I'll actually have you go through the task because it's a fairly complicated task. Even though once you understand it, there's not many different things being presented to it. So afterwards, we replayed these scenarios and then presented this number line literally. So the end of each number line is 0 and 10. And that's why we had them give a rating between 1 and 9. And we put one of those 3 individuals in the middle where 5 would be. And then we place a novel individual, and it was a novel individual every trial, somewhere on that number line between the boundary, which was fixed and always 0 and 10, and the participant and the rating of the friend or themselves, whoever's initials it was, or the Mr. or Mrs. Normal. That's what MN would be. And the key aspect of learning this novel individual's rating on each trial was knowing it was always relative to whoever in the middle this anchor exemplar's rating was. So although it was always put at 5 in a relative sense, you needed to have absolute coding. So if your probability of having a meeting with a foreign coworker was actually a 9, this would be between 0 and 9% proportionally, and this would be 9 to 10. So you have to mentally rescale that. Does that make sense to everybody? All right, and feel free to ask me questions. And so once they inferred that rating, so if, let's say I was a 9 for having a meeting with a coworker, this new person's probably like a 1 or a 2, you'd say. So, okay, so is this new person, then it would be a forced choice task with whoever wasn't in the middle. So is this new person closer to my friend or Mr. Normal, if those two people weren't in the middle? And then the only difference in the other two conditions would be that the friend would be in the middle, and you'd be learning it relative to the friend and relating to whether this novel individual is closer to Mr. Normal or yourself, or with this canonical or typical individual, Mr. Normal in the middle, whether that was closer to yourself or your friend. And this allowed us to, and subject could do this in a self-paced, forced choice manner, even though we gave them a max of 9 seconds, because they usually responded within 4 seconds after being trained, and they could go through each of these scenarios for each person in the middle once. So there's over 300 trials. And so this is kind of what we were hoping schematically they had an idea of where it was, even if they didn't have a precise idea. And although we tried to get them to choose a friend that was different, obviously there'd be some overlap with some of the preferences, and there's significantly more between the friend and themselves. But we didn't count those trials, and we just told them if it was, like, let's say these two people are both 5, either answer is correct. So here's the task. Let's have you guys give some ratings. You don't have to say it out loud. You can say it out loud if you want, depending on how enthusiastic you are this morning, 9 a.m. talk. Probably didn't make people too enthusiastic based on how much coffee they have. So what is the likelihood of you sending a message using mostly emojis? And this is on from 1 to 9, since 0 and 10 are the boundaries. So that way we can still present a novel individual at the extreme. So you have to choose between 1 and 9. And what do you think of the typical person of the same gender? What's the likelihood of them sending a message using mostly emojis? Oh, and your friend. Choose a friend that's different than you, that has a very different personality than you. Give that same rating. All right, everybody have that? Good. All right. So now we have a typical trial. So sending a message using mostly emojis. We have this new blue guy, and it's kind of halfway between 10 and whatever the rating you gave for the typical person. All right, so has everybody figured out what this blue guy is based on your response to the typical person? All right, so would that new person be closer to you or your friend?
Friend.
All right. So you got that correct. Congratulations, Rob. And so cognitively, what we think is going on here is we want people to mentally rescale where it is and then discriminate between the choices. And so here's just another example. And so this gave us, for the 3 conditions, this gave us 3 regressors of interest. First, we used a behavioral model, which I can go into depth at the end of the talk if people are interested. But it was just looking at choice discriminability of those options that you're choosing between based on the ratings. So just the difference, so you just have the difference between, essentially the model is I think I have an animation for this. So yeah, essentially the model is the difference between these ratings and the target individual. And so the smaller the difference, the more entropy or the higher uncertainty or the less discriminable it was. And since it was also informed by confidence, which correlated with their distance error, you could see like a normal distribution here. If for B, whoever B was in this case, had very low confidence on that rating, that might take up most of the scale, and that would be a more ambiguous decision versus if you knew that for sure. And so that all feeds into one regressor, just we call choice entropy, but it's really just the discriminability. And then the second factor that we looked at is rescaling. So how far towards the periphery was the actual rating that we gave for the person that was in the middle in order to infer the novel agent's rating. So that's just the absolute value. So ratings of 1 to 9 that were placed in the middle that would be a lot of mental rescaling versus ones that were in the middle, there'd be barely any rescaling. So yeah, that's a good animation I forgot I had. So here, this would be a more ambiguous choice, a less discriminable choice, whereas this more far away, there'd be lower uncertainty and it would be much easier to discriminate. And then with rescaling, if we had to shift it, if, let's say, sending a message using mostly emojis, I put myself as a 1 or a 9, then that would be more rescaling versus if it was a 4, 5, or a 6. And then the last thing was just whether they answered it correctly or not, so if there's some other factor that we couldn't explain by these two things that we modeled. Does that all make sense? All right, so just to illustrate the ratings. So for a typical individual, not surprisingly, Mr. and Mrs. Normal had a normal distribution around 5, as you can see in the turquoise, whereas the friend was more evenly distributed through 1 through 9 through these 100 different scenarios, with the self being slightly more discretized around 1, 5, and 9, but not too much different than the friend. And showing that they really learned this and that they had a firm idea of this social knowledge is that, and this even surprised me a bit, is that on over 70% of the trials, they were within 1 of the rating they gave beforehand. So for these other people, so they were highly consistent. And they only had catastrophic errors of more than 3 on different places on the number line for less than 10% of trials. So I was really happy about that I could, because it's not many moments in like a memory task or an episodic memory task where you can probe that many different elements of memory, not have them completely confused, and also have this stable representation, or what people at the Rotman would say a stable schema of probing different schemas. And as I alluded to earlier, just there was more overlap between the self and the friend, so we had to throw out more trials versus canonical with the friend, and self versus canonical individual. Ignoring overlapping ratings in this forced choice task, the participants performed about 72.5% of the choices correctly. So they were well above chance, but this was still a very difficult task. But they were able to infer it fairly quickly, answer an average trial within 4.7 seconds. And that the confidence ratings correlated with their subsequent consistency of their ratings, as I alluded to earlier. And then with our behavioral model, so since it was informed by confidence, this is just saying choice difficulty. The most difficult choices were when they had to choose between the canonical and friend. This was just because they had less confidence about their ratings for the typical individual and their friend versus themselves, where they obviously have more confidence. So that's all that's really saying here. And that's something to keep in mind when I present interactions by condition. So just looking at choice, just the main effect of choice discriminability across all trials. When we find it's a very ambiguous choice that's hard to discriminate, the 2 peak regions that we observed in this task were the retrosplenial cortex, where the peak was, and also the hippocampus. And so this is corresponding to when it was close together, which is almost reflective of a pattern separation process, when you need to disambiguate these 2 like ratings that you already had solid social knowledge of. What we notice, and these didn't significantly differ between the conditions based on who was being compared, although you will notice, and this will be important on the next slide, that the self comparisons it's mostly, of course, the hippocampal response is mostly corresponding to the self comparisons. So when you're sampling personal knowledge in the choice process, along a continuous dimension, or fine-grained social knowledge, I guess I refer to it. But then, more interestingly, and we also saw it extend a bit into subiculum and entorhinal cortex. But what we found was a hippocampal subicular entorhinal, because we didn't have very high resolution in the study, dissociation, where entorhinal subicular choices, like the hippocampus, signal these fine-grained differences for self-comparisons. But that it was a negative correlation with the choice difficulty when the self comparison wasn't involved, when they're comparing the novel individual to the friend and the typical individual. So that's really interesting. And this negative correlation instead of a zero correlation, what you see with the hippocampus, I think that's the main take-home difference. And I wouldn't say that the entorhinal cortex mainly represents other people more discreetly than the self, or only does that. So like, let's say, I'd say if for this task, if they had even confidence for their ratings about all the individuals, including themselves, we only tested things that they were sure about, I would say this would probably be a positive correlation. But luckily for me, it afforded a dissociation between the hippocampus and entorhinal cortex. That the entorhinal cortex and subiculum are these areas that can represent knowledge at multiple levels of complexity, whereas the hippocampus, at least in this task, is only along fine-grained one level of resolution or complexity, would be probably the take-home message from here. And just looking at rescaling, this is a great sanity check for me, is just like the areas with parietal neglect, you see that the peak is in this bilateral superior parietal lobule. When you had to rescale the anchor exemplar from the middle to the periphery in either direction. And you also see lateral orbital frontal cortex, areas associated with behavioral change. And another interesting thing that I didn't plan on, or didn't predict at all, was that along with the retrosplenial and subiculum activity effects that I found, that also predicted trial-by-trial performance, whether they got it accurately or not. So not only were they disambiguating these stimuli, they related to successfully disambiguating the two options. But more surprisingly was with the ventral striatum, since we didn't have any feedback, they just had that one thing on the screen to do, and then it was ITI, and then it was another choice. There was no feedback or anything, or no other element of the task. We saw that the strongest correlation with performance that wasn't predicted at all by choice discrimination or this rescaling was in the ventral striatum, and which increased prior to correct choices. And we were thinking, okay, so if the choice discriminability and the amount of rescaling doesn't explain what's driving the accuracy, what was the ventral striatum doing here? So what we thought was, given the role of the importance of the striatum in landmark versus boundary-based navigation and more procedural learning, we thought maybe when participants were decomposing these decisions into when the target could just be broken down into a high or low judgment. So if you infer that the novel agent is like a 1 or a 9, for the most part in that task, you could just say, who's lowest for who's least likely to send a message using emojis if the novel agent's 1, or who's most likely to eat spicy food if the novel agent rating was 9. And so we broke up trials into trials where the target agent was between the two options versus where they were from the side to the side of one of the options. And what we found was we also found a peak in the nucleus accumbens and ventral striatum for these high-low judgments, and that the participants, so this correlation here that has poor resolution, is the subjects that performed these high-low order judgment trials much better. The more they performed that better than the basic discrimination when they're between the two, showed stronger ventral striatal activity too, suggesting that might be the sort of procedural landmark strategy, even though this is on a one-dimensional number line, so when you're kind of aligning yourself to highest or lowest, you see more striatal activity during this process. So just to summarize, we tested a transformation of relational knowledge during social decision making. And we find that discriminating between different familiar individuals' preferences resembles a pattern separation process in the hippocampal formation and retrosplenial cortex. And that the entorhinal cortex and subiculum can signal knowledge at different levels of resolution for different reference frames. And that striatal signals reflect ordinal judgments, at least in this task. So I'm really interested by this because I think it highlights a potential division of labor between the hippocampus and entorhinal cortex, or within the hippocampal formation if we start to focus on hippocampal subfields, representing personal knowledge at multiple levels of complexities during social learning. And just the take-home messages overall from this talk is that spontaneous neural firing in the hippocampus relates to place cell reactivation and also selective default network fMRI signal increases. And that the hippocampal theta rhythm might reflect updating a location within a decision space. And that distinct reference frame computations within the hippocampal formation during knowledge-guided decision-making can be observed. So now to focus on how Lethbridge can facilitate my future research. So I think my research program contributes to a couple of specific research interests at the CCBN. First is the functional organization of long-term memory. And you can kind of see that of how I think knowledge is represented in just how the structure of how we encode things and retrieve things and how that might influence decisions. And then also the contribution of, I changed, edited a bit, to contribution of frontal hippocampal regions in decision making, but also, as you can see, as much as I try to avoid the basal ganglia, it always creeps in somewhere. And then also this computation approach to biological information processes. So looking at metric and ordinal coding. This is a phenomenon related to behavior that we can look at multiple levels, at the cellular level, and we can also look at the fMRI level and test this in animals and in humans. And I'm interested in coming up with normative accounts that can be tested at all levels for that. And then also just cognitive neuroscience and neuroimaging. Looking and exploring with fMRI, looking at different hippocampal subfields and brain areas, but also having comparative EEG or intracranial measures as a comparison. I feel like here not only is it a great opportunity to look at neuroimaging, but really to do translational neuroscience from cells to systems and develop hypotheses at the rodent level and test that at the human level and vice versa, and have this interchange between the multiple levels, especially with the classic focus here on higher level cognition in rodents, and see how we can really translate that to humans. And just a flavor of the experimental paradigms that I'd like to test in the future is which elements of autobiographical memory inform learning and decision making. So here I enforce knowledge, these ratings on 1 to 9 scale, but are we sampling from a schema? Are we just taking the most salient episode to give that rating? What's informing that rating? And what occurs during that sampling process? And then also, as I alluded to earlier, metric versus ordinal coding and learning decision making in both rodents and humans. And another question, which is really important and really a tough thing, and Matt can allude at the level of robotics and artificial intelligence, is generalization, and especially during on-the-fly planning or decision-making in novel situations. How we generalize from experience in one environment and use that to make a decision in a novel environment, a completely novel situation. And that's still very difficult for cognitive neuroscience to. And lastly, just given the pre-existing strength here, especially with circuit level measurements, just really looking at endogenous influences on memory encoding and decision making in rodents and humans, and seeing what motifs can help us understand that at multiple levels. So I would just like to thank all the collaborators on this work, and I was lucky that all this work was funded by the Wellcome Trust and allowed me to travel a variety of places without having one specific supervisor, although I must thank primarily Carl Fristing and Gustavo Deco for helping with this work, especially helping me conceptualize the computations that I was really, for the behaviors that I was really interested in. I have time for questions, and thanks for coming.